For much of the 20th century, the story of human evolution appeared increasingly settled. Modern humans were understood to have emerged in Africa and, over tens of thousands of years, expanded across the globe. Along the way, they encountered other human groups who had evolved earlier in Eurasia. Denisovans were assumed to be a distant eastern cousin, barely known and quickly gone. This picture felt orderly. Each population had its place. Each followed a separate evolutionary path. When contact occurred, it was thought to be rare and biologically insignificant. Neanderthals researched it even less. In fact, for most of modern science, Denisovans were not recognized at all. Until very recently, everything known about them came from a handful of remains, a finger bone, a few teeth, all discovered in a single cave in southern Siberia. From this limited evidence, Denisovans were described as a small, isolated group, living on the fringes of the human story. With no widespread fossils and no clear archaeological culture, they were treated as an evolutionary curiosity rather than a population that mattered. This interpretation shaped how human ancestry was explained. Denisovans were mentioned briefly, if at all, and usually dismissed as genetically irrelevant to modern populations. The assumption was simple: if Denisovans had played a meaningful role in human history, there would be clearer physical evidence, more sites, more bones, more tools. But this assumption depended on a fragile premise. In tropical and subtropical regions, heat, moisture, and soil chemistry destroy bone quickly. Entire populations can leave almost no skeletal trace. Even if they once numbered in the tens of thousands, absence of fossils does not necessarily mean absence of people. In the early 21st century, a new source of evidence began to challenge these long-held ideas. Ancient DNA made it possible to study human history without relying solely on visible remains. Genetic material, preserved inside bones and teeth for tens of thousands of years, offered a direct record of ancestry, contact, and inheritance. When scientists began comparing this ancient DNA to the genomes of living people, Denisovan genetic signatures appeared far from Siberia. Embedded within populations across Asia and Oceania, these discoveries quietly undermined the traditional view. Denisovans were not distant, marginal, or insignificant. They were deeply connected to the story of modern humans. To understand how such a hidden influence remained invisible for so long, we must first return to the assumptions that shaped human evolutionary thinking before DNA rewrote the evidence. For most of the 20th century, interpretations of human evolution relied almost entirely on coal, fossils, stone tools, and site locations formed the backbone of scientific understanding. From these materials, researchers reconstructed migration routes, population boundaries, and evolutionary release. It was a careful process, but also a limited one. Shaped by what happened to survive in the ground within this framework, human history was organized. Neanderthals were associated with Europe and Western Asia. Earlier hominin species were tied to specific regions and time periods. Each group was defined largely by skull shape, bone thickness, and tool styles. Cultural differences were often assumed to reflect biological separation. Denisovans entered this picture very late, and almost by accident. When the first Denisovan DNA was extracted from a small fingerbone in Denisova cave in 2010, researchers were surprised to find that it did not match Neanderthals or modern humans. But without a skull or a clear archaeological tradition, Denisovans remained difficult to interpret. They were classified as a sister group to Neanderthals, living somewhere in Asia. But little more could be said. From an archaeological perspective, Denisovans were almost invisible. Outside of Denisova cave, no sites could be confidently attributed to them. Stone tools found across Asia were typically assigned to either modern humans. There was no obvious Denisovan suke, no distinctive burial practices, and no recognizable art or symbolic behavior that could be clearly linked to them. Because of this, Denisovans were often treated as a geographically restricted group, mainly in Siberia or nearby regions, adapted to cold environments much like Neanderthals. Their apparent absence from Southeast Asia, New Guinea, and Australia reinforced the idea that they played no role in the ancestry of populations living there today. Traditional models of human migration further supported this view. According to these models, modern humans left Africa in one or two major waves, spreading rapidly along coastlines. As they moved, they were thought to have largely replaced earlier populations rather than mixing with them. Interbreeding, if it occurred at all, was considered rare and evolutionarily insignificant. This replacement-focused narrative left little room for Denisovans to matter. Without fossils in most of Asia, and without visible cultural traces, they were assumed to have disappeared long before modern humans arrived in many regions. Any genetic contribution they might have made was expected to be minimal. Yet even before ancient DNA became widespread, cracks were beginning to appear in this tidy picture. Archaeologists working in Southeast Asia and Sahul noticed long gaps in the fossil record. Human occupation appeared suddenly in places like Australia, with little evidence of gradual movement. The speed and complexity of these migrations raised questions that tools and bones alone could not fully answer. By the early 2000s, it was becoming clear that archaeology had reached the limits of what it could reveal on its own. The story of human evolution was more complex than regional fossils suggested, but without a new kind of evidence, that complexity remained largely hidden sips where physical remains could not. The breakthrough came from a set of techniques that were once considered impossible. Ancient DNA research began with a simple but radical idea: that genetic material could survive long enough inside bones and teeth to be recovered tens of thousands of years later. Early attempts in the 1990s were unreliable, often contaminated by modern DNA. But advances in cleanroom procedures, SUNEC technology, and computational analysis gradually transformed the field. By the late 2000s, researchers learned how to extract DNA from the dense inner portions of bones, especially the petrous part of the temporal bone in the skull and the inner layers of teeth. These areas protect genetic material from heat, moisture, and microbial damage. Even so, ancient DNA is highly fragmented. Most samples contain only tiny pieces of genetic code, mixed with environmental DNA, precaria, and fumbia, millions of short sequences, and comparing them against reference gum. What made the Denisovan discovery possible was not just extraction, but comparison. When scientists sequenced DNA from the finger bone found in Denisova cave, they compared it to known Neanderthal and modern human gums. The result was unexpected. The Denisovan genome was distinct from both, yet clearly related. At first, this finding seemed limited in scope. One cave, one individual. But the true impact emerged when Denisovan DNA was compared to the genomes of living people around the world. Drake noticed something striking. Certain populations carry genetic variants that could not be explained by Neanderthal admixture. These Denisovan genetic signatures appeared most strongly in populations from Melanesia, Papua New Guinea, and parts of Australia. In some groups, up to 5% of their genome showed Denisovan ancestry. This was far higher than anything seen from Neanderthals outside of Europe and Western Asia. Even more surprising, traces of Denisovan DNA were also found at lower levels across East Asia, South Asia, and parts of Southeast Asia. This pattern raised immediate questions. How could a population known from a single cave in Siberia have contributed so much DNA to people living thousands of kilometers away, many of them in tropical regions where Denisovan fossils had never been found? The only explanation was repeated contact and interbreeding between Denisovans and migrating modern humans. As more ancient humans were sequenced, the picture grew even more complex. Denisovans were not a single uniform group. Genetic evidence suggested deep internal diversity, with Denisovan populations separated from each other for hundreds of thousands of years. Some Denisovan lineages diverged early. Modern humans appear to have encountered different Denisovan groups at different times. Dating these events relied on molecular clocks, which estimate when populations mixed based on mutation rates. The results suggested that Denisovan admixture occurred multiple times, beginning shortly after modern humans left Africa. This was not a single encounter. It was a long process of interaction. Perhaps most remarkably, Denisovan DNA was detected, even where Denisovan bones were absent. In some cases, genetic material was recovered directly from cave sediments without any visible fossils at all. This confirmed that Denisovans occupied regions where their skeletal remains had not survived, reshaping assumptions about their true range. By the mid-2010, Denisovans from a genetic curiosity into a central figure in human evolutionary history. The challenge was no longer proving their existence. What the DNA ultimately revealed was not a minor episode of contact, but a deep and uneven pattern of genetic exchange that reshaped how scientists understood Denisovans and their role in human history. Instead of a single isolated population, Denisovans now appear to have been widespread, diverse, and regionally adapted across much of Asia. One of the most important findings was that Denisovan ancestry is not evenly distributed among modern populations. People from Melanesia, particularly in Papua New Guinea and nearby islands, carry the highest proportion, often between 3 and 5%. Such a high percentage cannot be explained by a single brief encounter. It implies sustained or repeated interbreeding between modern humans and Denisovans. In East and Southeast Asia, Denisovan ancestry is present at lower levels, but it is widespread. Genetic studies show that populations in China, Southeast Asia, and parts of South Asia carry distinct Denisovan-derived segments that differ from those found in Melanesia. This indicates that modern humans encountered at least two, and likely more, genetically distinct Denisovan populations as they moved across Asia. These findings directly challenged earlier assumptions. The traditional model suggested that modern humans largely replaced earlier populations with minimal mixing. Denisovan DNA shows instead that interbreeding was common enough to leave a lasting biological legacy. Modern humans did not simply pass through Asia, the populations that had lived there for hundreds of thousands of years. Ancient DNA also revealed that Denisovans themselves were deeply structured. Some Denisovan lineages split from others more than 300,000 years ago. This means that when modern humans encountered Denisovans, they were not meeting a single group, but multiple populations that had long separate histories. One of the most striking discoveries involved genes related to environmental adaptation. Certain Denisovan-derived genes affect immune responses, metabolism, and adaptation to local conditions. The most well-known example involves high-altitude adaptation of foliated. This genetic contribution appears to have provided a survival advantage in high-altitude environments, where modern human physiology alone would have struggled. This finding reshaped how scientists think about admixture. Interbreeding was not simply a biological accident. In some cases, Denisovan genes were retained because they offered immediate benefits. Natural selection favored these variants, allowing them to persist and spread within populations. Ancient DNA also revealed evidence of population bottlenecks and extinctions, despite their wide range, appear to have declined and disappeared as distinct populations over time, likely contributed to their disappearance. But extinction did not mean erasure. Their genetic legacy survived within modern populations, especially in regions where Denisovans had lived for long periods. Perhaps most importantly, these discoveries forced scientists to reconsider the geographic boundaries of ancient populations. Denisovans were not confined to Siberia. Genetic evidence play fossil preservation is poor. Their apparent absence from the archaeological record was not evidence of absence, but a limitation of preservation and discovery. By revealing these patterns, ancient DNA transformed Denisovans from a marginal group into a major contributor to modern human ancestry, but of interaction, mixture, and shared survival across deep time. The consequences of these discoveries extend beyond genetics and into how we understand humanity itself. Ancient DNA has shown that modern humans are not the product of a single isolated lineage, but the result of repeated encounters between closely related populations. Denisovans were not outsiders to this process. They were participants in it, shaping the biological makeup of millions of people alive today. One of the most important shifts has been the realization that human ancestry is shared in complex and uneven ways. Denisovan genetic contributions differ by region, reflecting local histories rather than a single global event. This means that modern human diversity cannot be explained solely by recent migrations or cultural differences. It is rooted in deep evolutionary interactions that occurred tens of thousands of years ago. These findings have also reshaped how scientists interpret adaptations certain Denisovan derive in environs. High-altitude regions, pathogen-rich tropical environments, and new ecological zones pose challenges to incoming populations. Yet exchange provided tools for survival that would otherwise have taken many generations to evolve. Importantly, this does not suggest superiority of one population over another. Instead, it highlights cooperation. When modern humans encountered Denisovans, interbreeding allowed useful traits to move between populations. Over time, these traits became part of shared human biology, blurring the boundaries between groups once considered separate species. The Denisovans no longer exist as a distinct population. They are not entirely gone. Their DNA continues to influence immune responses, physiology, and adaptation in living people. In this sense, extinction does not mean complete disappearance. It can mean transformation and integration. For anthropology, the currid, what? Chure. It reminds researchers that absence of evidence is not evidence of absence, especially in regions where climate destroys fossils quickly. At a broader level, Denisovan ancestry knows a shared human past. Modern populations across Asia and Oceania carry traces of ancient encounters that link them to people. These connections are not symbolic or cultural. They are biological, written into the genome. By revealing these deep links, ancient DNA shifts the narrative away from division and replacement, and toward continuity and connection. Humanity did not emerge fully formed from a single source. It was shaped through contact, movement, and mixture, leaving a legacy far richer and more complex than previously imagined. The story revealed by ancient DNA is quiet, but profound. It shows that the history of our species cannot be fully understood by bones and tools alone. For most of human existence, populations lived, moved, and interacted in ways that left little visible trace. Denisovans were one of those populations for thousands of years. Their presence was hidden, not because they were rare or unimportant, but because the environments they inhabited did not preserve their remains. It provided a record that does not depend on survival of skeletons or artifacts. Denisovans re-entered the human story, not as a mystery population defined by absence, but as a widespread and influential group whose legacy remains alive today. Their DNA shows that interaction, not isolation. These findings do not replace earlier archaeological knowledge. Instead, they complete it. Fossils and tools show where people lived and how they behaved. DNA shows who they were connected to and how those connections shaped future generations. Together, they reveal a human past that is layered, regional, and deeply interconnected. The Denisovan contribution to modern ancestry also serves as a reminder of how much remains undiscovered. Entire populations may still be invisible in the fossil record, known only through the genetic traces they left behind. As techniques improve and more ancient DNA is recovered, the human family tree will continue to change, becoming less like a simple branching diagram and more like a network of overlapping histories. What emerges from this research is not a story of replacement or Denisovans. Denisovans did not vanish without impact. Their influence persists in physiology, adaptation, and survival, carried silently through generations. Ancient DNA does not provide final answers. It offers a clearer window. Through that window, Denisovans are no longer shadows at the edge of human history. They are recognized as contributors to the shared ancestry of humanity, reminding us that our origins are more complex and more connected than we once believed.